Hello friend, welcome in Pharma Talks. In the series of videos of analytical method development for chromatography, it is the second part. In the first part, I am discussing about what is the procedure, what is the scope, what is the purpose of the method development, and what is the procedure, what initially we have to do during first start the method development. Now, along with these, I am going to start it. What can we do next during the method development procedure, because we have learned what is the initial parameters or initial thing we can do during the method development. That is we have to perform the literature search. With the help of literature search, you have learned the physiochemical properties, their fit log value, their pKa value, their solubility, their polymorphism, their storage, all. And along with these, now we are going to discuss next. Okay, here we have to start. So in the current scenario during the when we perform the method development, analytical target profiling to preparation is must, because everything in the pharma sector, it should be compared, or it should be compared like a quality by design, or some compliance system should also introduce in the R&D sectors. So it is also helped me both much. So analytical target profiling is very important topic. So ATP, what is the ATP? ATP is the analytical target profiling, is the summary of quality requirement of a method. An ideal profile should consider all requirement of a method. It should be classified into three categories. So before reading the categories, it should be categorized in table 1. Okay friends, yes. The first requirement of ATG is the method's perspective. So what is the method perspective as per the ATP? So in the method's perspective section, we have to compare these some parameters. So the first one is the method with adequate resolution between the peaks of interest. It means the resolution should be greater than 1.5 for the peak of interest. With high detection sensitive, means S by N ratio should be greater than 10 at the reporting threshold level. A good peak shape to ensure accurate quantification means USP training less than 2, theoretical plate should be greater than 2000, and the stability indicating power is also required. And the second perspective for the method ATP, analytical target profiling, is regulatory perspective. What is means by regulatory perspective? So your specificity. Okay, regulatory perspective means specificity. The method you have to develop, it should be specific, it is linear, it is precise, accurate in the desired range. Other regulatory requirement like percent RSD for SA as per USP Journal Chapter 621, and other requirement of ICHQ2 USP recommendation. Okay, it should be comes under the regulatory perspective. Business perspective means the method you develop, it is a simplified procedure for user and environmental friendly. It is a simplified procedure for user environmental friendly. High throughput, less chromatographic runtime, means the method you develop, it should be a less runtime, and sample preparation time should be less. It can save the time, and the time is the money. Affordable, your method should be affordable in the term of cost and durability of columns, availability of equivalent column in the market, cost of solvent, reagent, etc. Presence of method which can be executed with available resources before demanding any special resources. Your method should be robust and rugged. Performance of method with respect to day-to-day system analyst variation and ability to tolerate small variation duration during execution. Preference of single method like same column mobile page for all tests like RSSC and resolution. It should be preferable and good. The third topic, the third topic we must know before for the method development technique, selection of analytical technique. Okay, friends. So selection of analytical techniques should be based on several considerations such as available literature and method, physiochemical properties, and available analytical resources. Chromatographic. Wait a minute. Okay. For non-chromatographic, volatile and thermally stable compound, GC is preferred technique. However, for such compound, by converting to a stable, non-volatile chemical derivative, other techniques such as LC, UV spectrophotometry should be attained. It should For liquid chromatography, please refer to chart 1 given at the end for further guidance. Method development for liquid chromatography. These are the step by step procedure. If you have to follow these all procedure, you should easily develop analytical method for a compound. Means some hindrance or you also face, but you will easily develop. This is applicable for related substance assay, content analysis, content uniformity, dissolution, etc. The formulated sample would be received from the various department location like FRD, outsourced parties, manufacturing sites for method development. The approach 1, method development for drug product or drug substance using available references, that is pharmacopoeial references, reported literature or any other references are present. The first approach is, if you have a literature search, if you perform the literature search and you find some literature which can have a method, it should be a pharmacopoeial, non-pharmacopoeial or any other. So what you have to do, in the case of literature reference method, it is important to evaluate the suitability of reported method for the drug product or drug substance and make necessary change if it does not meet the requirement of ATP. And what is the requirement of ATP? It should be 3 requirement which was we discussed earlier. The second approach is comes in our mind during the method development is the method development for the drug product or the drug substance in the absence of adequate literature and references. In the absence of enough literature or in case of existing method are not suitable for the dosage form, method development or API, method development should be started from the fundamental by considering the physiochemical properties of the analyte and ATP. Method development and optimization should be initiated preferably with the help of software like ACD, Design Expert, etc. Conventional approach can be adopted based on prior knowledge and experience. Column selection. How you select the column for your first analytical run? For reverse phase chromatography, the choice of column stationary phase can be made on the following parameter and their suitability for the current application. First parameter is the particle size of the silica. Second one, functional group of polymeric layers that are attached to the silica surface. Method development should preferably be initiated with C18 or C8 column. So it is a thumb rule, not any written. If you start the method development, if not any general things you found in your structure or any specific functional group or anything in your during the literature, so you should started with the C18 or C8 column. Carbon loading, you should know on the carbon loading. Surface area, column length and internal diameter, detail of end capping and type of end capping material, column to column variability, concern of pH range in which column packaging material is capable of remaining unaffected. In general, a column can be used in the pH range of 2 to 8 of mobile phase. However, based on requirement, special columns are available that can be used when the pH is outside the above range. So, types of column. Types of column. C18 column, rugged, widely used polar compound, can be retained well when compared to a column with shorter carbon chain. C8, similar to C18, polar compound can be retained well when compared to the column with shortened carbon chain. C3, C4, less retentive, less stable, used mainly for peptides and proteins. Phenyl and phenethylene columns, moderately retentive, selective, can be useful for the separation of compound with closely related aromatic structure. Use of pi-pi interaction for the separation. Cyano column, moderately retentive, normal phase. It is a column which can be used in both reverse phase and normal phase. Amino column, weakly retentive, more often used for normal phase, less stable. This type of column can also be used in both reverse phase and normal phase. Polystyrene column, stable for pH between 1 to 13. It is a good peak shape and lifetime. Selectivity change often found less efficient. Helic columns, for the separation of the polar and ionic compound, use hydrophilic stationary phase with reverse phase type element. Normal phase chromatography. Okay, friend, this is enough. We have read, we have know in the part second of the method development video. In my next part, we are going further and discuss what should the criteria for to develop or we should have to take in during the normal phase chromatography and what perspective in my mind. If you like the video, please share with your colleagues and share and subscribe my channel for more interesting video. Thanks for a lot for watching this series of video. Thank you.